Hi everyone. So my name is Anika and I work as a senior researcher in GANOS at the group for epigenetics led by Professor Vlatko Zoldos, and I'm also affiliated with the University of Zagreb Faculty of Science. And today I'm going to talk about our efforts to try to understand the IgG glycosylation a bit better. So for those of you who've been here last year, I'm going to repeat myself quite a bit in the first 15 minutes, so you can take a quick nap. And for those of you who are here for the first time, I really hope this will be interesting. So, as you all know, glycans can really affect the function of a protein. And this is especially true for IgG. And IgG has this conserved end-glycosylation site on the Fc region, or the effector region of the antibody. And the presence of glycans can really affect how IgG functions in the immune system. But not only the presence of glycans, but also the composition of glycans on IgG. And Manfred talked about core fucosylation. For an example, afucosylated IgG will have increased ability to activate ADCC, but also other glycan structures on IgG will also affect how it functions and will fine-tune the immune response. So it's not that surprising that IgG glycosylation changes in different pathological and physiological conditions. And here on the left you can see all of the different diseases in which IgG glycosylation is changed. You probably can't see because the letters are too small, but different autoimmune diseases, different neurological disorders, also cancer, different physiological conditions like, for example, increase of body mass index like obesity, menopause, and so on. And here you can see our data that we gathered in GANOS in the last 15 years that spans 34 different cohorts and all of these different conditions, so ranging from autoimmune diseases like lupus, ulcerative colitis, rheumatoid arthritis, and so on. We also have data from allergy, from chronic obstructive pulmonary disorder, also different CVD events, type 2 diabetes, only one cancer, colorectal cancer, and we have 4 aging cohorts. And when you look at these graphs, you can see that the pattern of changes are somewhat similar. We mostly see that glycans that lack galactose increase in all of these different conditions, and ones that have 1 and 2 galactoses and that have sialic acids decrease in most of the conditions. But for some, there are some disease-specific changes. For example, in heart failure, you can see this increase of core fucosylation. And when you inspect galactosylation a bit better, you can see that in all the disease, most of the diseases and aging, glycans with 2 galactoses are decreased. And the ones that have 1 galactose are actually decreasing most of the diseases, but they remain quite stable in aging. So there are some disease-specific mechanisms, and we really gathered a lot of data about how glycans change in different conditions. And we know that they impact the function of IgG. But one thing we know very little about is actually how glycosylation of IgG is regulated. So protein glycosylation in general happens in the ER and the Golgi with the help of all of these different enzymes. So glycosyltransferases, glycosidases, transporters. What is also important is the availability of sugar donors and of course cellular contacts and protein sequence, so the accessibility of the glycosylation site. But what we were interested in is actually how is glycosylation of IgG regulated specifically. Which of these mechanisms are most important for IgG glycans? Is it the expression, activity, and localization of this glycosylation machinery? Are the sugar donors that's important, or is the accessibility of glycosylation sites? And we actually started to gain a lot of insight from genome-wide association studies. But actually, they left us with more questions than answers, because most of the genes, we actually didn't know how could they be implicated in the process of glycosylation. Just some of them were actually part of this glycosylation machinery. And relatively recently, we performed largest GWAS of the human IgG glycome up to date. It was done on more than 20,000 samples, and we discovered 42 genome-wide significant loci, and were able to prioritize 83 candidate genes. And again, most of these genes are not implicated in the glycosylation of proteins either directly or indirectly. So we had no idea how they could be involved in this process. And we had to come up with a way to test this. And we decided to use CRISPR technology. So the approach is pretty straightforward. First we thought maybe we could use knockouts and classical CRISPR, but we knew we wanted to better mimic what happens in the organism, because these SNPs certainly do not knock out these genes. They probably maybe change the expression of them a little bit. So we opted for transcriptional regulation. And what we do is we use CRISPR to either activate or silence the candidate genes, and then we analyze how this intervention actually affects IgG glycosylation. And just a quick overview how this technology works. So it makes use of this dCas9 protein, which is catalytically inactive Cas9, so it cannot bind to DNA, it cannot cut DNA or induce double-stranded breaks, but it can bind guide RNAs. And guide RNAs can be synthesized to be complementary to any sequence of interest. And guide RNAs will then guide this dCas9 protein to the sequence of interest. And if you fuse any type of effector domain on dCas9 protein, you can actually do different interventions on that DNA sequence. And what we actually use VPR and CRAB. So VPR is a transcriptional activator, and when you guide a VPR to a promoter of a gene, it will activate its expression. And CRAB does the opposite, so CRAB will actually lead to heterochromatinization and consequently gene silencing. And all of the CRISPR tools we use in our lab are based on this modular CRISPR dCas9 toolbox. And it's a really cool system because it enables us to assemble any type of cassette that we want to use in our cells really quickly. So it relies on golden gate cloning method. So we can have that one-step assembly of different modules. And here are the modules that we currently have in our lab. So our toolbox supports guide RNAs. We can have up to 11 guide RNAs, so we can target up to 11 different genes, or we can target one gene with many guide RNAs. Also, we have different promoters. We have weak ones, strong ones, also some tissue-specific ones. And then comes this fun part, and these are the effector domains. So I talked about VPR and CRAB, but we also have different domains that will target specific epigenetic marks. So we can directly modulate DNA methylation, also different histones modifications like methylation, citrullination, acetylation, and so on. We also have this different dCas9 orthologs. Currently, we are developing the miniaturized version of these that are just that are smaller, maybe threefold than the ones we have now. And also, we have different selection and screening markers and transcriptional terminators. And we also expanded the backbone, so we can deliver our CRISPR tools transiently, but we can also deliver them stably to our cells, either with transposon technology or with lentiviral technology. And using this system, we actually established this first generation CRISPR dCas9 platform to try to validate, to functionally validate all of these different GWAS candidate genes. And you can see here we had to adapt the system just a little bit. So we decided to integrate our CRISPR tools into the genome of the cells. But you can see there is no guide RNAs here. So our tools are silent in the cells. And when we are ready to target a GWAS candidate gene, we just place guide RNA on the same plasmid that contains genes that code for IgG. And this design makes everything so much simpler because we don't need any type of selection, because IgG secretion will happen only in the cells where targeted manipulation took place. And then we did a proof of concept experiment. You can see here how IgG glycome looks like in our cells. So these are HEK cells, so we chose them because they do have the glycome that is quite similar to the one we see in human plasma. All the main structures are present, but those that were low in abundance were really hard to quantify. And for the first experiment, we decided to target these different glycosyltransferases that are both involved in the process of glycosylation, but there were also GWAS candidate genes for IgG glycosylation. And manipulation of all of these genes actually resulted in expected changes of IgG glycome phenotype. So we knew that the system could be used for this purpose, and that CRISPR actually works in this way. And then we were ready to target different GWAS candidate genes. And actually, we were pretty successful in manipulating most of them, but maybe around 1/4 of manipulations actually resulted in IgG glycome changes. And here you can see some of them. These are the results from this estrogen study that Professor Lautz already talked about. So this was a really cool study where, from prior studies, we know that women, when they enter menopause, their IgG glycome significantly changes. And in this study, we actually had healthy women that were put on hormone suppression therapy, so they had induced menopause, and then they were randomized to receive either placebo or estradiol. And in the placebo group, glycans dramatically changed. So the same thing happened as in the natural menopause. But in the estradiol group, actually, the glycans remained completely stable. So estradiol prevented those changes from happening, giving us proof that estrogen really could be regulating IgG glycosylation. And then we scanned through our GWAS candidate genes to find the ones that are estrogen regulated, and we found quite a bit of them and manipulated some of them. And for these two, we actually observed specific changes of IgG glycosylation. But you can see that they are not the same. So when we upregulated this transcription factor, what we observed is decrease of both glycans with 1 and with 2 galactoses. So this is the same phenotype we see when we silence B4-GALT1, glycosyltransferase that adds galactose. But when we upregulated SPINK4, we actually observed only a decrease of glycans with 1 galactose, pointing to two specific mechanisms that regulate mono and degalactosylation. And then we focused a little bit on genes that were associated with IgG galactosylation specifically, and we observed the same thing. So for manipulation of some of them, we did observe only changes in glycans with 2 galactoses, and for some only with 1 galactose. And at this point, we could only focus actually on, as you can see here, on glycans with galactose, because those other structures were really hard to quantify. So we had to come up with a way to be able to see how these changes affect other glycan structures also. And what we did, we developed this second generation platform. So we did two things. So we first introduced Fab glycosylation site, because Fab glycans are always a bit more complex than FC glycans. And you can see here that this chromatogram is much more complex than the one that I showed. And you can see that there are many glycans with cellulation and bisecting both-up, which enables us to quantify them. But again, this made the problem of our cell system a bit more obvious, because we observed many glycans that are not native to IgG, some with GalNAc and antenary fructose, which made the calculation of the derived traits really hard, and also data interpretation was really hard. So we did another thing. We improved the resolution of our system. So we introduced codon-optimized sequences of IgG genes for the expression in human cells, and we also reduced the backbone size quite significantly to increase the delivery to our cells. And this actually resulted in a pretty improved resolution and enabled us to quantify those low abundance glycans on the FC also. And just as I established this system, this really interesting story popped up, where we tried to see how obesity affects IgG glycosylation. And you can see that in obesity, both glycans with galactose and sialic acids are decreased, and this is accompanied with really significant alterations of B-cell transcriptome. And we scanned through these differentially expressed genes and found these two, which are actually mapped to the same SNP, together with all of these different other genes that we prioritized. And then we tried to see their expression of all of them with qPCR, and we observed that VNT3 was significantly upregulated in obesity. And then we did the same thing in mice, so we had obese mice, and again we observed that VNT3 was really upregulated in mice. So we put our new platform to the test, to see if we can replicate these changes, and we upregulated VMP3, and we actually observed a decrease of both sialylated and galactosylated IgG glycans, so the same phenotype we see in obesity. And what we know is that VMP3 plays an important role in obesity, but how it affects IgG glycosylation, we still don't know. And we figured out that we could use this platform for another mechanism that is also very important for glycosylation, and that is protein structure. So we are always quick to say that addition of glycans is a non-templated process, but the truth is protein structure really constrains glycosylation. So what we did, so we know now that there are more than 50 AG alleles in humans, so there are many different IgG allotypes. And these allotypes can change the function of antibodies, but nobody actually looked at how these different allotypes affect IgG glycosylation. And we had some hints from this collaborative cross-mice study, where we found that two IgG allotypes had really distinct MC-linked glycoprofiles. And you can see it here. So on the position 296 of IgG1, if you introduce isoleucine in place of phenylalanine, you can see a big decrease of both galactosylation and sialylation. And there was one really old paper that observed that if you introduce alanine on the same position on IgG3, again you get a big reduction in galactosylation and sialylation. So we mutated IgG1, so position 296, and human IgG1 has tyrosine on this position, and we mutated to alanine, phenylalanine, or isoleucine. And you can see here that we actually confirmed the results both from mice and from that really old paper. So here is the rate of galactosylation for phenylalanine, and that is that allotype of IgG found in mice. And here you can see a really big decrease when we change the amino acid, and also a big decrease with the addition of alanine. And what is really interesting is that human IgG3 has two most frequent allotypes, and these are these two. So one with tyrosine and one with phenylalanine. And you can see there is a big difference. And we know that these two allotypes can change the antibody function, and maybe that the cause of it is glycosylation. But let's get back to the CRISPR studies. So we always wanted to change the cell line because we knew we had the wrong cell type with HEK cells, so we wanted to transfer the technology to B cells, but that wasn't that easy because our CRISPR cassettes are enormous and they are really hard to deliver to the cells of interest. And I screened through many different B cell lines and somehow landed on this ARC77 B cell line, which is actually a lymphoblastoid cell line. And you can see how IgG glycome looks like so in these cells, so it's much more similar to the human plasma IgG glycome. Only these structures that are primarily come from the Fab site are missing because this IgG does not have Fab glycosylation site. And we established our system in the ARC77 B cell line. So the first part, the integration of our VPR and CRAB tools is completely the same. So we obtained monoclonal cell lines that express either VPR or CRAB, but without guide RNAs. And the cool thing here is that we were actually able to select high-producing clones that produce a lot of IgG, and this enables us to really easily quantify IgG glycosylation, which makes everything so much easier. And then we had to come up with a way how to deliver guide RNAs to our monoclonal stable cells, and we had to use again transposon technology to integrate guide RNAs, so we had enough time to actually gather IgG. And so we placed guide RNAs between ITRs of Sleeping Beauty transposase. And Sleeping Beauty transposition it's a similar technology to piggyback, but it does not interfere with piggyback, so everything works together. And this worked pretty well. And I decided to test the system on that same gene, VNP3, that we upregulated in the second generation platform. And you can see here that we were successful in upregulating VNP3, and we observed the same changes of IgG glycome phenotype that we observed in our HEC cells. So both glycosylation and sialylation were reduced. And when I established this system, I really wanted to target one gene that was always interesting to us, and this is MAMBA. MAMBA is beta-aminosidase, and it's a lysosomal enzyme that is part of the N-glycan catabolism, so it's the final exoglycosidase, and it does this step. And we actually targeted this gene in the first generation system, and we observed changes of monogalactosylation only, which was quite interesting. And then we did the experiment again in the second generation system, but we could reliably quantify both alpha-1,6 arm and alpha-1,3 arm, so galactosylation on one and on the other, and we observed that they were both reduced, and nothing happened to degalactosylated IgG glycans, so they remained stable. And then we did the same thing in ARC77 cells, and something pretty unexpected happened. So we observed that only galactosylation on alpha-1,6 arm was reduced significantly. And this arm is preferentially galactosylated on IgG, and we actually now have this almost the same level of alpha-1,6 arm and alpha-1,3 arm on IgG secreted from these cells after we upregulate MAMBA. And we know that these two types of galactose can also change how IgG functions in the immune system, but we have no idea how MAMBA could be doing this because it is a lysosomal enzyme. We have some hypotheses, but these are really new results, maybe 3 or 4 weeks old. So we are trying to think of the ways how to test this. So you will probably have to come here next year to see if we came up with something cool. And that's all. I just want to thank all of my colleagues and friends who participated in all of these studies, and thank you all for listening.